unfortunately it is cached. Okay. It also, before I start, this is off the record. It occurs to me I've done this talk 3 times now, and I've only been absolutely sober one of them. Tonight is not one of those. Alright, let's see if I can get this right. Okay, my name's Dick Wall. I work for a company called Navigenics. I have a job that I am fortunate enough to absolutely love. I work with genetic scientists, and I find this stuff really fascinating. So I'm going to give you a quick rundown on a high-level overview of genetics and genetic risk calculation. So 101 genetics. DNA is organized into chromosomes. Chromosomes have genes. Human beings have about 25,000 genes. That number used to be thought to be a lot higher. The scientists have pretty much isolated it to 25,000 now. And genes contain these things called SNPs. So what's a SNP? A SNP is a single nucleotide polymorphism. It's, I guess, a fancy word for a mutation. In English, a DNA is a phrase, or DNA phrase, where a nucleotide, there's 4 nucleotides, A, C, T, G, they can vary between 2 possible states in different people. So an example here is ACTCCTAG in one person might be ACTGCTAG in another. And these changes can be statistically significant for increased risk of a health condition. So, genetics 101. You probably remember this from school, if you did human biology. You get 2 copies of your DNA, one from each parent. For a SNP, you've got 2 possible allele types. That's the risk and the non-risk allele. So times 2 copies, you get 4 possible combinations. You get risk-risk, risk-non-risk, non-risk-risk, or non-risk-non-risk. For the purposes of this calculation, risk, non-risk, and non-risk-risk are collapsed together. We can't really tell the difference for those kind of numbers, so there is some risk involved in that. And these are the common names: homozygous risk, heterozygous risk, and homozygous non-risk. Okay. Genetic composite index, this is where the fun math part starts. So in some ways this is a bit like bookies giving odds on a particular horse race or something like that. Basically scientists take these different SNPs that you have and they assign odds ratios to the risk, homozygous risk, heterozygous risk, or homozygous non-risk. The odds ratio is normalized to 1.0 for non-risk, so the others will naturally be higher. They're a greater risk. The product of all the odds ratios gives you this thing called the GCI score. In itself, it's a meaningless number. But if you take the GCI for a person, divide it by the GCI for the average population, and then multiply it by the average estimated lifetime risk of a condition, you get a person's estimated lifetime risk. So there's a lot more to this. This is a very, very simplified version of the calculator we use. But it actually makes a pretty interesting case for functional programming. So I've got 2 minutes. Great case for functional. Basically the process goes like this. So if you can see here, you start with the SNP, which will have 2 alleles. The alleles in this case are A and G. That will map to risk, non-risk, and that will map to an odds ratio of, let's say, 1.8171. The GCI, you take that, you multiply it by the other risk markers for that condition, and you get a result. It's a number, but this is the largely meaningless number until you put it into context. Then you take that number, you divide it by the average population, you multiply it by the risk, the estimated risk for the entire population, and you get that person's risk. So, by example here, let's say we've got a condition that has a 35% occurrence in people, in the average population. Our GCI score is somewhat higher than the average, so the result comes out as a 42% lifetime risk for this person. And some of you may notice that this is actually a map-reduce operation. So the first part, taking the alleles, mapping them through to the genotype, and then mapping them through to a number, that's a map. The multiplication of them all together is a reduce operation. So now I'm going to skip ahead a little bit. This is actually a talk that is normally much longer. And Joel wanted some Scala, so let's get a bit of Scala up here. Man, this is really small. Parallel arrays, Scala, okay. So this is how the actual calculator roughly looks in Scala. Man, it's really small. Let's talk on tomorrow afternoon, and I can show you all of this, but these lines of code are effectively the core of a simplified version of the genetic risk calculation we use at our time.